This section explains how to study a single DNA lesion inside a living cell. Damage a genome with anything real, and you get a heterogeneous smear of adducts, untractable because you cannot ask what any single lesion does when 100 coexist. The move is to collapse the mixture to one variable. You synthesize a genome carrying one defined lesion at a known site, then let the cell itself be the readout. Bypass efficiency measures whether the polymerase survives the lesion. Mutation frequency and specificity measure what base it guessed opposite. Swap and repair, deficient host strains ADA, OCUL, and ALK-B knockouts, and the same probe now isolates which pathway erases which damage. The primitive is general. Convert an uncontrolled chemical population into a controlled biological experiment by making the input singular and the organism the instrument. This section explains how a single defined lesion in a viral genome becomes a measurable causal variable. Put exactly one chemically defined lesion at a known site in a viral genome, pass it through a living cell, and you convert an intractable problem into a single variable causal assay. The intractable version is this: cells hit by damage yield a heterogeneous smear of adducts, none individually attributable. None individually attributable. The defined site version replaces that smear with one lesion you can trace. The chapter's real move is ratiometric. Never score the lesion genome alone. Co-transfect it with a near-identical unmarked competitor, so transfection variability becomes common mode noise that the ratio divides out. Replication blockage reads as a drop-in lesion to competitor output. This is the Crab assay. Mutation reads from the same population. Swap the host for a repair-deficient strain, and that same ratio attributes which enzyme acts on which lesion. The lesion is the independent variable. The competitor is the internal standard that makes it measurable. This section explains base excision repair and the portable recipe that its opening enzymes represent. Base excision repair opens with a glycosylase, an enzyme that recognizes one lesion and snips the damaged base from the backbone. The family is redundant by design, with overlapping specificity so no single failure leaves a lesion unrepaired. FPG and OGG1 read oxidized purines. The endonuclease 3 and endonuclease 8 line reads oxidized purines. The endonuclease 3 and endonuclease 8 line reads oxidized pyrimidinines, and Mujwai excises the 8-SOXO-G. The chapter's real object is the biology only on its surface. Its true object is a portable recipe. Clone, overexpress, affinity purify, then assay on a defined, damaged substrate, deliberately abstracted from any one enzyme. That is its function in the book. When a freshly sequenced genome yields a candidate glycosylase, you already hold the template to make it a working reagent and measure what it recognizes. This section explains what a DNA glycosylase is at the level of its structural primitive. A DNA glycosylase is its substrate specificity. It is a hydrolase that recognizes one class of damaged base, flips that base out of the helix, and severs the glycosidic bond, initiating base excision repair. That is the whole entity. Name the lesion, it reads, and you have named the enzyme. This chapter reads as a reagent recipe rather than a record of discovery. Sequencing surfaces, new glycosylases faster than anyone characterizes them, so you keep the known bacterial and human homologs as bench standards, FPG, nth, eth, ni, mui, and their human counterparts. Each newcomer is then defined by comparison against them. Recognition is deliberately redundant. Overlapping specificities are the cell's backup, and the assay inherits that same partition. Oxidized purine, oxidized pyrimidine, mispaired adenine. This section explains how a second family of DNA glycosylases was discovered through the absence of a defect. A knockout that changes nothing is a promise. Something parallel absorbs the loss, OGG1 and NTH1. No mice accumulate oxidized bases yet stay healthy, so mammalian cells must run a second, unseen class of oxidized base, glycosylase. The chapter locates it by homology. It runs a blast search of the human genome against the bacterial prototypes, Ni and SPG, and two orthologs precipitate out, Neal1 and Neal2. What marks the class is mechanism. An N-terminal proline nucleophile drives beta-delta elimination against the plain beta elimination of the Nth and OGG1 lysine. That one difference predicts the rest. A 3' phosphate end needing PNK rather than APE1, and an appetite for single-stranded, bubble, and actively transcribing DNA. Redundancy was first sensed as silence, then named by its bacterial echo. This section explains what makes one repair glycosylase distinct from another, and why that single distinction ripples through the whole pathway. A repair glycosylase is individuated not by which damaged base it flips out, but by the chemistry its active site nucleophile runs on the backbone, and that one choice propagates through the entire pathway. NTH1 and OGG1 use an internal lysine, run beta elimination, and hand a 3' phosphominoester to APE1. Neal1 and Neal2, the mammalian Neal and FPG orthologs this chapter purifies, use an N-terminal proline and run beta-delta elimination. They leave a 3' phosphate that APE1 barely touches, so they must recruit PNK instead. The tell that they existed at all. OGG1 and NTH1 no mice accumulate lesions yet stay healthy, which demands a redundant activity. Their signature is a preference for single-stranded and bubble substrates, repair aimed at DNA caught open, mid-transcription or mid-replication. This section explains how one oxidative lesion sabotages its own repair. A lesion is what it does to the machine that tries to remove it, and that is the whole chapter compressed. 2. Deoxyribonolactone, abbreviated DL, is an oxidized abasic sugar. It enters base excision repair normally. The enzyme APE1 incises it, exactly as it would an ordinary abasic site. Then the pattern inverts. The next enzyme, DNA polymerase beta, would normally excise the incised sugar with its lyase activity. Here it becomes covalently trapped on the sugar instead, a suicide substrate that turns the repair enzyme itself into a DNA protein crosslink. The cell survives only by rerouting to long patch base excision repair, resynthesizing 2 or more nucleotides to bypass the poisoned step. The damage is defined by the trap it sets for its own repair, rather than by its chemistry. This section explains how a single damaged DNA base can turn its own repair enzyme into a hostage. The lesion is called 2-deoxyribonolactone, written DL for short. It enters base excision repair the ordinary way. The enzyme AP1 nicks it just as it would normally release the enzyme, so the catalytic attack freezes into a covalent crosslink between the DNA and the protein. Does not excise the sugar phosphate stub the way it should. Instead, it attacks the sugar phosphate stub the way it should. Instead, it attacks the stub with its lysine nucleophile, and then it cannot let the elimination step that would normally release the enzyme. So the catalytic attack freezes into a covalent crosslink between the DNA and the protein. The repair step becomes a suicide bond. This is the mechanism-based inhibitor pattern, a suicide substrate, except it arises in place inside the genome rather than being designed as a drug. The cell's only exit is to reroute the damage onto long patch-based excision repair, which resynthesizes straight past the trap. This part explains how a single oxidized base gets caught by the third enzyme in a repair relay. Oxidation turns guanine into 8-oxo-G, a lesion that still reads as a legal partner. Adenine pairs with it happily, so replication inserts A opposite the damage, and the mismatch machinery sees nothing wrong. MUT-E, the bacterial enzyme, and its eukaryotic homologue, MYH, answer this by excising the base that is chemically correct but genealogically wrong. They remove the misinserted adenine, not the lesion, betting that resynthesis restores cytosine and hands the damaged strand back to the guanine repair enzymes. Remove the lesion instead, and you would fix the mutation into the genome. MUT-E's target choice is the whole trick. It is the third guard in a relay against one oxidized base, and its failure predisposes to colorectal cancer. This section explains why the repair of oxidized guanine takes 3 enzymes, and why the third one cuts the base that looks correct. Oxidation turns guanine into 8-oxo-G, which mispairs with adenine. The defense is not 1 enzyme, but 3, staged against a single lesion. MUT-T scrubs the oxidized nucleotide from the pool. MUT-M excises 8-oxo-G, paired with cytosine, and MUT-E, this chapter's subject, excises the adenine after misreplication has already seeded as. The move worth holding is this. MUT-E removes the undamaged base. Error is defined relationally. Adenine is innocent alone, wrong only opposite an oxidized partner, so repair keys on the pairing, not the lesion. Cutting the correct-looking base buys 1 more replication round to correct the mismatch, before a G to T, C to A transversion fixes it permanently. This section explains how an invisible kind of DNA damage becomes measurable the moment you build a cell that dies of nothing else. AP sites, the abasic lesions left by spontaneous depurination, form at roughly 10,000 per mammalian cell per day, yet they leave no phenotype in a healthy genome. Too many redundant repair routes mask them. Strip those routes down to 1. The APIN1, APIN2, RAD1 triple mutant can no longer clear its endogenous AP sites, and it survives only as microcolonies teetering at the edge of lethality. Now viability itself is the readout. Cross in a 4th mutation and watch the colony. Smaller, larger, or unchanged, and every gene sorts by whether it feeds the AP site burden, repairs it, or ignores it. The sensitized background is the instrument. Push a process to the brink of lethality, and perturbations of survival report on it. This section explains why the cell must constantly repair abasic sites, and how deleting that repair turns colony size into a way to measure invisible damage. Endogenous abasic sites accumulate faster than intuition allows, roughly 10,000 per mammalian cell per day, from the bare hydrolysis of the bond between base and sugar. Their repair is therefore not maintenance, but a condition of living. The chapter's move is to make that invisible load legible by breaking its repair. Delete APIN1, APIN2, and RAD1 through RAD10 together, and yeast dies. The triple mutant survives only as microcolonies. Colony size then becomes a dosimeter. Cross in a 4th knockout and read the interaction. Genes whose loss shrinks the colony were sharing the repair burden. Genes whose loss rescues it were feeding the lesion pool. Synthetic lethality titrates a damage flux that no one can count directly. This section explains why DNA polymerase beta became the reference model for base excision repair. 1 enzyme carries 2 chemically unrelated reactions on 2 physically separable domains. That is the reason pol beta became the reference model for base excision repair. The 31 kilodalton polymerase domain runs nucleotidyl transfer, filling a single nucleotide gap. The 8 kilodalton amino terminal domain runs a lyase that eliminates the 5' deoxyribose phosphate left behind after AP endonuclease incision. 2 of the 4 coordinated steps of base excision repair thus collapse into 139 kilodalton protein, the simplest cellular polymerase with no proofreading exonuclease. The generative move is decomposition. Each mechanism sits on its own foldable domain, so each can be excised and assayed alone, and the kinetics resolve cleanly. Modular chemistry begets modular measurement. The enzyme's tractability is a structural consequence of its architecture rather than a lucky convenience. This section explains how one small enzyme comes to own 2 of the 4 chemical steps in base excision repair. Repairing a single abasic site takes 4 chemical steps, and one enzyme owns 2 of them. It owns them because it is 2 enzymes fused into one. DNA polymerase beta is the smallest cellular polymerase at 39 kilodaltons, and it cleaves cleanly into 2 pieces. One piece is an 8 kilodalton lyase domain that strips the DRP backbone remnant. The other is a 31 kilodalton domain that fills the 1 nucleotide gap by templated synthesis. 2 chemically unlike reactions. 2 active sites, 1 folded donut clamped on the gap. The decisive move is that this structural separability makes each activity independently assayable. That is why polymerase beta became the reference system for reading polymerase mechanism at all. Modularity in the substrate is inherited as modularity in the catalyst. This section explains how AlkB repairs alkylation damage in DNA. Most DNA repair works by cutting. It excises the damaged base and resynthesizes it from the intact strand. AlkB does the opposite. It leaves the backbone untouched and simply peels the illegitimate group off in place. A methyl group stuck on the ring, nitrogen of adenine, giving 1-methyladenine, or on the ring nitrogen of cytosine, giving 3-methylcytosine, blocks base pairing, AlkB is an iron 2, alpha-ketoglutarate dioxygenase. It hydroxylates that methyl, and the unstable hydroxymethyl falls away as formaldehyde, restoring the native base, restoring the native. Repair here is subtraction. The base is mended by taking something away with no excision at all. Here is the generative move. When the damage is an added adduct instead of a broken bond, the cheapest fix is to strip the adduct off in situ, with no cutting and no template needed. This works only where the lesion is graftable and exposed in single-stranded DNA at replication forks and transcription bubbles. This part explains AlkB, an enzyme that repairs DNA by reversing chemical damage directly instead of cutting it out. Most DNA repair excises the lesion and resynthesizes the strand. AlkB does the opposite. It erases the chemical modification in place, restoring the original base without ever cutting the backbone. A methylated adenine or cytosine, 1-methyladenine or 3-methylcytosine, is a carbon lodged where a hydrogen bond should form. AlkB, an iron 2, alpha-ketoglutarate dioxygenase, oxidizes that carbon and lets it drop off as formaldehyde, regenerating the native base untouched. The generative primitive is direct damage reversal, repair that undoes the lesion rather than replacing it. It acts only on single-stranded DNA because the complementary strand shields the alkylation face in a duplex. The toxic lesion exists only where the helix has transiently opened, at replication forks and transcription bubbles. This section explains how DNA photolase repairs UV damage and why it is a different kind of repair enzyme. Most repair enzymes replace damage. They cut out the lesion and resynthesize from the intact strand. Photolase instead reverses the chemistry that made the lesion. Ultraviolet light fuses adjacent pyrimidines into a cyclobutane ring, and the enzyme splits that ring back to two canonical bases, restoring the original sequence rather than rebuilding it. This is the whole primitive of direct repair, and it hands you a second surprise. The enzyme binds the dimer in the dark, but it cannot act until it absorbs a blue photon. Light is a substrate. Recognition and catalysis are thereby decoupled in time. You can trap the bound complex, then fire a flash and watch the bond splitting unfold from femtoseconds to milliseconds. The chapter is that mechanism, purified. This section explains photolase, an enzyme that repairs DNA damage by reversing it rather than cutting it out. Every other repair pathway cuts the lesion out and resynthesizes the gap. Photolase does the opposite. It reverses the damage in place with a blue light photon as the reagent. It binds an ultraviolet fused pyrimidine dimer in the dark, then absorbs a photon that injects an electron into the strained cyclobutane ring, which splits back to two canonical bases. The enzyme dissociates untouched. Two chromophores divide the labor. A folate or diazoflavin antenna harvests the photon, and a reduced FAD does the chemistry. The whole surprise is that this catalysis is a bis-substrate reaction whose second substrate is light. That is also why the same fold, stripped of its lesion, survives as cryptochrome, a pure light sensor. This section explains why deamination assays measure the scar instead of the reaction itself. You cannot watch an enzyme turn cytosine into uracil. The edit is a single silent loss of an amino group, so you measure the scar rather than the act, coupling the deamination to a downstream reader that tells uracil from cytosine. In the cell, the reader is selection. The deaminase mutates ARPO-B, the C to T change survives rifampicin, and you delete UNG, the glycosylase that would erase the uracil before it registers. In the tube, that same glycosylase becomes the instrument. It cleaves at uracil, and a shifted gel band counts the edit. One move, two substrates. Route, an invisible chemical transformation through a step that makes it countable, then strip whatever repair silences the signal. This section explains how an enzyme too small to watch is read off the damage it leaves. A deaminase converts cytosine to uracil. Unrepaired uracil copies as thymine, a C to T transition. Kocher's chapter turns that chemistry into a phenotype. Express the deaminase in E. coli, let it mutate the ARPO-B gene, and score the survivors on rifampicin. The mutation is the readout. The move that makes it work is subtractive. Delete UNG, the uracil glycosylase, so the base excision pathway stops erasing the signal before it can be counted. That is the generative primitive. To measure an invisible act, wire its product to a survival marker and knock out whatever repairs it. Sequencing the survivors then recovers the enzyme's fine specificity, which cytosines, in which context. This section explains how the two recognition factors of global genome repair find damage without ever reading its chemistry. Recognition here is indirect. Neither sensor reads the lesion's chemistry. Both read the geometry that the lesion imposes on the double helix. The XPC complex binds a junction between double-stranded and single-stranded DNA, the local unpairing that any helix-distorting adduct creates. So one structure-specific factor covers chemically diverse damage, with no need for a lesion-specific receptor for each kind of damage. UV-DDB is the specialist that plugs XPC's blind spot. UV photo products distort the helix too little for XPC to catch them alone, so DDB2 and DDB1 bind those lesions directly and hand them off to XPC. A UV-triggered ubiquitination of both factors couples detection to release. Recognition works as a timed switch, a grip that lets go on schedule rather than a static hold. The generative move is this: build a universal detector by sensing the invariant consequence, the distortion, rather than enumerating every possible cause. Then patch the low signal cases with a dedicated second sensor. This section explains how global genome nucleotide excision repair recognizes damage without ever cataloging the chemistry of the damage. The problem is one of enumeration, and the cell solves it by refusing to enumerate. The XPC complex never reads the lesion's chemistry at all. It reads the helix distortion the lesion induces, binding the single-stranded to double-stranded junction character that any bulky base damage produces. One structure-specific factor that any bulky base damage produces. One structure-specific factor, therefore, covers chemically diverse lesion causes, while it stays blind to the chemical defect itself. Some UV lesions distort the helix too little to trick XPC. Those get a dedicated pre-sensor, UV-DDB, which binds them directly and hands them off. Here is the twist that makes the chapter go. Recognition is a regulated bind rather than a static one. UV-DDB carries a ubiquitin ligase, and that ligase ubiquitilates both XPC and DDB2, tuning the handoff between them. The pattern to carry forward is this: recognize the class by its shared structural signature, and let a specialist patch the members the signature misses. This section lays out the structural claim of the chapter. Two protein lineages that share no common ancestor converge on one and the same geometry. The bacterial system over ABC and lift the damage out as a short oligomer. The human system uses 6 factors: XPA, RPA, XPCTF2H, the XPF2H, the XPFERCC1 complex, and XPG. Neither system shares an ancestral protein with the other, yet both bracket a lesion with 2 incisions, one on either side, and lift the damage out as a... That convergence is the chapter's structural claim, and the method is its proof. You reconstitute the nuclease from purified parts, then interrogate it with DNA you built to a known sequence and a defined lesion. The radiolabel is placed so that the excised fragment reports the cut. 3 assays: incision, excision, and resynthesis decompose the pathway into observable steps. The deep move is this: the recognition subunits can barely tell damaged DNA from intact DNA. Specificity comes instead from kinetic proofreading. A TP hydrolysis is spent to convert energy into information about the state of the DNA. This part explains how one repair operation reaches the same design twice by 2 unrelated routes. Cut out the damage, resynthesize, ligate. That 4-step operation logic. These DC incision logic by 3 subunits called over ABC, and in humans by 6 factors: XPA, RPA, XPC, TFII, XPC, TFII, XPC, TFII, XPG, and XPF-ERCC1. These 2 systems share no evolutionary ancestry, yet they converge on the same dual incision logic. The chapter's compression is what supplies specificity. The recognition subunits bind damaged and undamaged DNA almost alike, so discrimination cannot come from affinity. Instead, each assembly step burns ATP, and that dissipation is kinetic proofreading, the energy of hydrolysis converted into information about DNA structure. The rest is method. How to express each factor and reconstitute excision on a lesion at a defined site. So the abstract mechanism becomes a bench assay. This section explains how one enzyme's decision becomes readable when every other actor is removed. An enzyme's decision is only legible once nothing else can make it for you. Put a single defined lesion at a known site on the transcribed strand and let purified RNA polymerase II meet it with none of the initiation machinery present. No promoter, no TBP, no TF2B, no TF2E, no TF2F, no TF2F. The oligodicetane is the trick. It is a single-stranded handle. The polymerase loads onto directly, substituting for the whole promoter, plus factors apparatus. What remains is the bare choice. Stall at the lesion, which gives a short 130 nucleotide transcript, the signal that recruits transcription-coupled repair, or bypass it, which gives a full-length transcript, possibly mutant. Transcript length becomes a binary readout of that choice. The pattern. To attribute a behavior to one actor, delete every other actor that could co-produce it. This section explains what happens when RNA polymerase, not the replication machinery, meets a damaged base. Damaged studies default to the replication fork, DNA polymerase, copying a lesion. But the transcribed strand is assaulted too, and the reader, RNA polymerase II, faces the same fork in the road. It can stall at the lesion, summoning transcription-coupled repair or dying if none comes. Or it can bypass the lesion, misincorporating a ribonucleotide, exporting a ribonucleotide, exporting a mutant transcript, a souse that never divide. The chapter's move is subtractive. It uses an oligo-DC tailed template, carrying one lesion at a defined site, read by purified RNA polymerase II with no promoter and no general factors. Strip every confound, and the raw enzyme lesion encounter is all that remains to read out, the binary choice isolated as a phenotype. This section explains why every assay in the chapter reduces to a single move. Repair that prefers one strand can only be seen by an assay that tells the strands apart, so the whole problem collapses to one procedure. Convert an invisible lesion into a countable break, resolve it by strand, and read repair as the rate at which breaks vanish. The counting works in two steps. A lesion-specific nuclease cuts wherever damage sits. Then a strand-specific probe counts the survivors. A southern blot resolves fragments up to about 25 kilobases. Primer extension in the form of LMP-CR resolves down to a single nucleotide. Transcription-coupled repair is just the differential between the two strands of an active gene. The transcribed strand clears faster because a stalled RNA polymerase flags the lesion for repair. Everything else is resolution. The assay ceiling fixes which asymmetry, at which length scale you are able to detect. This section explains how one detection method makes DNA damage readable on a gel. The idea is simple. A lesion is invisible to a gel, so the whole method turns it visible by converting every lesion into a strand break. Once damage is a break, it becomes a change in fragment length you can read. The conversion is done by a lesion-specific nuclease. For pyrimidine dimers, this is T4 endonuclease V, and it nicks only at the damage. Damaged strands therefore run shorter, and as repair proceeds, the full-length signal recovers. Everything downstream is one choice of readout on that same break. A southern blot averages over a whole gene fragment. It is robust but low resolution. Ligation, mediated PCR, written LMPCR, reads single nucleotides. It is sensitive and position. resolved. Transcription coupled repair falls straight out of this. Probe the transcribed and non-transcribed strands separately from one sample, and you watch repair favor the strand being read. This section explains how a single enzyme, TFIIH, does the same physical act for two different jobs. One machine, two jobs, the same act. TFIIH is a 10 subunit complex carrying exactly 3 catalytic activities, 2 ATP-dependent helicases, XPB and XPD, plus 1 kinase, CDK7, both transcription and nucleotide excision repair. Recruit that same enzyme to do the 7 repair. Recruit that same enzyme to do the same thing. Pry the duplex open. Add a promoter, it melts the start site so RNA polymerase II can fire, and it phosphorylates the polymerase's CTD to release it into elongation. At a lesion, it unwinds around the damage so the strand can be dually incised. The compression is this. Repair and transcription share one machine, reused across contexts. The tell is pathological. Because the subunits are shared, mutations in XPB, XPD, or P8 scatter across 3 diseases: xeroderma, pigmentosum, Cockayne syndrome, and trichothiodystrophy. This section explains why a single molecular machine, TFIIH, serves 2 completely different jobs. It is one engine that unwinds DNA, called by 2 different callers. T-if is defined by what its 2 ATP-driven helicases, XPB and XPD, actually do. They pry open a DNA duplex. Its 10 subunits matter far less than that single action. Both transcription and nucleotide excision repair are contexts that invoke that same opening. Promoter, melting for RNA polymerase II, and lesion flanking bubble formation for repair are the same primitive reused in 2 settings rather than 2 separate mechanisms. The chapter's whole apparatus follows from this. Purify the shared complex once, then read its action through a staircase of assays on immobilized DNA, first a permanganate footprint, then dual incision, then resynthesis. Because the engine stays constant, the sequential arrival and departure of its partners around it becomes visible. The 3 linked diseases prove that the subunits separate cleanly by function. This section explains an assay that studies ubiquitination of RNA polymerase II and of other proteins inside crude yeast extracts. The starting move is simple. When you cannot purify the machine, use the cell as the reagent. Ubiquitination needs 3 enzymes, acting in series, an E1 activator, an E2 conjugator, and an E3 ligase, together with ubiquitin and ATP. Reed and Svedstrup lacked purified versions of these for RNA polymerase II, so they made a crude yeast whole. The extract already holds every factor, so the reaction reconstitutes in vitro without isolating any of them. This leaves the substrate as the only variable. Swap RPB1 for another protein, and the same assay reports the ubiquitination of that new protein. The biology it serves is DEF1-mediated tagging and destruction of a polymerase, stalled at UV damage. This is the cell's last resort, taken when transcription-coupled repair cannot clear the lesion. This section explains how to catch a ubiquitination reaction when you cannot name the enzymes that carry it out. When you cannot name the E1, E2, and E3 machinery that ubiquitinates a protein, do not purify it. Make the crude yeast extract itself the enzyme, since it already carries every factor. Add tagged substrate and mite-tagged ubiquitin, incubate, then immunoprecipitate on the ubiquitin tag. Whatever survives is the conjugate. Genetics does the dissection that chromatography cannot. A deletion strain subtracts one candidate, and add back of that lone purified protein restores activity. This converts correlation into direct requirement. Loss of DEF1 abolishes RNA, polymerase II, ubiquitination, and restoring DEF1 brings the activity back. The chapter's move is general. When the responsible machinery is unknown or unpurifiable, let the intact proteome supply it, and let strain construction, not fractionation, isolate the factor that matters. This section explains how the reconstituted human mismatch repair system corrects a base-base mispair. Error correction needs an asymmetry to break. Given a base-base mispair, which of the two strands carries the error? The reconstituted human system answers with a single signal. A strand-specific nick marks the new strand, and everything else follows as directional consequence. Exo-1 hydrolysis from the nick toward the mispair with a fixed 5' to 3' polarity. The mismatch itself is the stop codon. Excision terminates once the mispair is gone with RPA present. The generative surprise is that one polarity-locked enzyme services a nick on either side. RFC and pCNA reorient the geometry. The clamp decides direction. The polarity of the nuclease is fixed and carries no directional information of its own. The minimal parts list is MUTS alpha, MUT-L alpha, XO1, RPA, pCNA, and RFC. That this list is sufficient is itself the claim. This part explains how mismatch repair solves a problem that has no local answer. When repair meets a mismatched base pair, the base is chemically flawless on both strands, and nothing marks which strand carries the error. There is no local signal to read. The cell supplies the answer non-locally as a single strand break elsewhere on the daughter strand, a borrowed asymmetry that stands in for newly synthesized. The whole reconstituted system exists to convert that distant nick into exonucleolytic motion aimed back at the mismatch, terminating there. The surprise the chapter isolates is this. One exonuclease of fixed 5' to 3' polarity services a nick on either side. A 5' nick, it walks naturally. A 3' nick, it walks only once. RFC and pCNA re-reference its direction. The error is its own stop signal. This section explains how a transient repair machine is characterized by engineering, the very defect it responds to. A transient molecular machine has no isolable form. It exists only as what assembles on its trigger. The repairosome is exactly that, the protein swarm that mismatch repair convenes on a base mispair. Mismatches cannot be induced inside a living cell, so you cannot pull the complex from lysate directly. You bait it. You build a heteroduplex carrying a defined mismatch plus a strand nick. Eukaryotic mismatch repair has no endonuclease, so it degrades only from a pre-existing discontinuity, and that same nick marks which strand is newly synthesized. Then U-affinity marks which strand is newly synthesized. The substrate is the reagent. The machine is operationally defined by its bait. You characterize the repairosome by engineering the defect it answers to. This part explains why studying mismatch repair forces you to build its substrate by hand. Mismatches cannot be induced inside a living cell, so the machine that hunts them can only be watched in vitro. That means you must manufacture its prey. This is the chapter's whole move. Build a defined heteroduplex, a small circular duplex carrying one deliberate mispair or extrahelical loop, plus a single nick, a known distance away. Anneal the linearized double strand of one phagamid to the single-stranded circle of another differing at one position and the defect sits at a chosen site. This engineered substrate is at once bait and assay. As bait, DNA affinity purification pulls out the entire mismatch repairosome. The nick is not incidental. It supplies the strand discontinuity that mismatch repair needs to decide which strand to degrade. This section explains how a mismatch repair protein doubles as a sensor that decides a damaged cell should die. The same protein that corrects a typo also decides the cell should die, and it decides by failing to correct. MUT-S-alpha recognizes a mismatch, and once it is loaded with ATP, it recruits MUT-L-alpha to excise the daughter strand. But mismatch repair always targets the newly made strand. When the offending base sits on the template instead, an alkylation adduct, say excision, removes the innocent copy, resynthesis reinserts the error, and the machine fires again. This is the futile cycle, repair that cannot succeed but cannot stop, each turn leaving a single-stranded gap. The accumulating gaps become the alarm. First, the ATR checkpoint arrests the cell, then P53 and P73 drive apoptosis. So a proofreader doubles as a damage sensor by the simplest trick, making its own inability to win into the death signal. This part explains how a single repair module flips into a damage sensor when its repair cannot finish. Mismatch. Repair corrects a wrong base by cutting the new strand, never the template. Hand it a lesion. The template itself carries, and correction can never terminate. MUT-alpha recognizes the mispair, excises it, resynthesis reinstalls the same error, and recognition fires again. This futile cycle leaves a single-stranded gap that never closes, and the unclosable gap is itself the signal. ATR activates. The cell arrests, apoptosis follows. So one recognition module reads as a corrector on a removable error and as a sensor on an irremovable one. Nothing changes but the substrate. The primitive to carry forward, a repair machine, becomes a damage detector exactly where its repair cannot succeed. The failure to terminate is the message. This part explains why E. coli tolerates DNA damage instead of repairing it, and how the cell keeps its error-prone tools under lock. Damage tolerance is distinct from repair. It is the cell choosing a finished, flawed genome over a stalled, perfect one. When a lesion blocks the high-fidelity replicative polymerase, E. coli licenses a normally caged error-prone specialist to copy straight across the block. Two such specialists carry this out: POL4, also called DINB, and POL4, also called DINB, and POL4, also called DINB. The generative primitive here is the licensed exception. A mutagenic tool is kept locked and unlocked only under proven damage. The lock is the SOS circuit plus a temporal switch. OMAD is cleaved to OMAD prime only slowly against a REC-A and single-stranded DNA filament, so the same protein first enforces a checkpoint, then later an A filament, so the same protein first enforces a checkpoint, then later enables bypass. Fidelity is spent deliberately to buy survival. The mutation rate is the receipt. This section explains why the polymerases that rescue a stalled replication fork are the same enzymes that introduce mutations, and why an elaborate control system must exist to hold them in check. A replicated polymerase stalls at a lesion because its active site demands a correct Watson-Crick pair. The Y-family polymerases, in Escherichia coli, the polymerase 4 enzyme also called DINB, and the polymerase 5 enzyme also called UMUD'2C, succeed at that lesion precisely by relaxing the demand. D'2C succeed at that lesion nuclease so it accommodates a damaged template. that same looseness misreads undamaged DNA. Lesion tolerance and infidelity are therefore one axis, not two separate properties. The enzyme that saves the fork is intrinsically mutagenic. The whole regulatory apparatus follows as a necessity. The SOS transcriptional response and the Rec-A timed cleavage of UMU-D into UMU-D prime, which withholds translesion activity until damage is confirmed. A dangerous fallback must be gated to its emergency. This methods chapter is the toolkit for dissecting that gate. It covers expressing the polymerases and the accessory factors that decide when they fire. This section explains how you measure the fidelity of translesion DNA synthesis when fidelity itself is invisible at the molecular scale. The assay makes the polymerase's own mistake write itself into a reporter gene. You let the enzyme copy a defined lesion embedded in the LAC-Z gene, drop the product into E. coli, and each error class collapses onto a single plaque color. A dark blue plaque marks a base substitution at the amber codon. A colorless plaque marks a base substitution at the amber codon. Counting the plaques, then sequencing them, turns an error rate into arithmetic. The generative move is this: engineer the substrate so that every mismatch maps onto a discrete visible channel. Measurement becomes selection. From there, you can predict the rest. Any lesion, any polymerase, any sequence context slots in unchanged, because the only thing that must be preserved is the mapping from error to phenotype. This section explains how Kunkel measures the errors a polymerase makes when it copies past a DNA region. Those errors are far too rare to read off the molecule directly, so the move is to make the cell's phenotype the meter. Kunkel's trick copies a LAC-Z template carrying one defined lesion, then drops the product into E. coli. An error that flips the amber codon turns a light plaque dark blue, and a frame shift turns it colorless. Sequencing those mutant plaques names each substitution, insertion, and deletion. The rare molecular misinsertion is amplified by biology into a countable, classified by biology into a countable, classifiable colony. The generative move is to couple an invisible molecular event to a selectable reporter phenotype. Unlike single nucleotide kinetics, this scores the complete bypass. Insertion plus extension with all 4 deoxynucleotide triphosphates competing as one error spectrum. This section explains why the polymerases that copy past DNA damage are built as the mirror image of the ones that copy undamaged DNA. A tight, active site makes a replicative polymerase accurate, and that same tightness is exactly why a bulky lesion stops it dead. The Y family translesion, polymerases invert that constraint. They have an open active site with an extra little finger domain, the PAD, no proofreading, exonuclease, and low processivity. The same geometric slack that admits a damaged base makes copying sloppy. Fidelity and lesion tolerance are one axis run in opposite directions. A cell buys damage tolerance with error. The chapter is the recipe. These enzymes are too scarce inside the cell to touch directly, so you express recombinant affinity tagged versions. Each one runs beside a catalytic dead twin, the same enzyme with its DE active site residues mutated to AA, which proves the activity is theirs and not a host contaminant. So O, which proves the activity is theirs and not a host contaminant. This section explains how a cell copies its DNA straight through a damaged spot. Replication stalls at a lesion because the high-fidelity polymerase demands a well-shaped template it no longer has. The cell's answer is not repair, but tolerance. It swaps in a specialist that copies straight across the damage, that is translesion synthesis, carried out by the Y family polymerases, whose active sites are held deliberately open, with an extra little finger called the polymerases, whose active sites are held deliberately open, with an extra little finger called the PAID. The trade is exact. These enzymes abandon proofreading, processivity, and fidelity to bicontinuity. The generative move is a division of labor by tolerance, a generalist that stalls, and an override enzyme that finishes the stretch and hands the fork back. Everything the chapter does, purifying these enzymes and deleting their genes in mice, is instrumentation built to watch that override at work. This section explains why purifying an error-prone polymerase means running its natural regulation in reverse. A cell that survives lethal UV does so by licensing a deliberate mistake. Maker, Pol5, the error-prone polymerase that copies past the lesions. Pol3 stalls on, trading fidelity for a replicable genome. The chapter's move is that purifying such an enzyme is the inverse of the machinery restraining it. Every step undoes a control. The cell evolved to keep Pol5 rare and safe. You pre-cleave UMU-D to UMU-D prime to skip the activating gate. You delete the chromosomal UMU-DC so no poisonous UMU-D. UMU-D prime heterodimer triggers LPXP proteolysis. You throttle expression to a weak promoter so UMU-C stays soluble. The protocol is the regulation. Read backwards. This section explains how a bacterium licenses its own mistakes in order to survive DNA damage. Survival buys itself with mistakes when an unrepaired lesion stalls the genome it rescues. Pol3 at the replication fork, E. coli deploys a deliberately sloppy enzyme, DNA polymerase 5, to copy straight across the damage. This copying across the damage we tolemeres, we called across the lesion, is called translesion synthesis, and it pays 100-fold rise in mutation for the genome it rescues. The mistake is licensed. Pol5 stays caged behind 3 gates. The first gate is SOS induction of the UMU-DC operon. The second is self-cleavage of UMU-D into its active form, UMU-D prime. The third is REC-A. REC-A plays an unusual role here. Pol5 needs it as a built-in subunit of the mutasome, the complex written as 2 UMU-D prime subunits, 1 UMU-C and REC-A. Rather than the nucleoprotein filament, REC-A forms elsewhere. The chapter's purpose is to purify that native heterotrimer intact enough to watch a cell mutate itself on purpose. This section explains how a cell keeps replication moving when its DNA is damaged and why a chapter on these enzymes is really a chapter on purification. Replication stalls when a lesion breaks, the Watson-Crick fit that the high-fidelity polymerase demands. The cell's answer is not repair, but substitution. It swaps in a low-fidelity Y-family polymerase whose roomier active site accommodates the damaged base, 1 specialized tool per lesion class. Each tool is matched to its damage. Pol-eta reads through sunlight, induced pyrimidine dimers accurately. Pol-iota flips the template purine into the syn conformation and pairs it by Hoogsteen geometry. Rev1 just inserts C opposite a basic sites. Pol-zeta extends whatever mispaired terminus the others leave behind. The chapter's craft is expressing these enzymes in Saccharomyces cerevisiae rather than Escherichia coli. It exists because you can only characterize a bypass polymerase. You can purify in milligram quantity. This section explains how a cell trades replication accuracy for the ability to copy past damage. Replication fidelity is something the cell swaps in and out, tuned to the moment rather than held fixed. When the high-fidelity replicase stalls at a damaged base, the cell recruits a low-fidelity Y-family polymerase shaped to that specific lesion, one specialist per damage class. Pol-ETA handles UV cyclobutane dimers. Rev1 handles UV cyclobutane dimers. Rev1 handles a basic sites. The inversion carries the chapter. These enzymes are error-prone on normal DNA precisely because their active sites are roomy enough to hold a distorted template. The same slack that makes Pol-ETA sloppy on clean DNA lets it copy a dimer accurately. Chapter 24 is the recipe express and purify each enzyme from yeast to read that specificity biochemically. Tolerance bought through disposable inaccuracy, swapped in only at the stall. This section explains how a stalled replication fork swaps polymerases by retuning affinity. When damage halts the high-stringency replicative enzyme, a single ubiquitin is welded onto pCNA, the sliding clamp every polymerase docks on. That lone covalent mark raises the clamp's affinity for the sloppy, lesion-tolerant Y-family polymerase, which then wins the docking site and copies past the lesion. Deployment is spatial and chemical at once. The specialized polymerases already wait inside replication factories, so the mark only has to decide which one grips the clamp. Predict the pattern anywhere. A covalent tag flips, which of two competitors captures a shared binding site. The modification carries the decision, and the scaffold merely obeys. This part explains how a stalled replication fork recruits its repair polymerase. A stalled fork does not summon the repair polymerase by sensing the lesion. It marks the clamp. When a replicative polymerase jams at damage, pCNA, the sliding ring encircling the DNA, gets monoubiquitinated. That single covalent tag raises its affinity for the low-fidelity Y-family polymerases, which then displace the high-stringency replicase and copy past the high-stringency replicase, which then displace the high-stringency replicase and copy past the lesion. The switch is affinity modulation on a shared hub. Nobody reads the damage directly. A mark on the common scaffold reweights who binds, and the enzyme swap falls out of that reweighting. The same primitive governs any regulated handoff where a post-translational mark on a docking platform resorts its clients. The platform decides. The tag is the vote. This section explains how Sugawara and Haber made a rare DNA repair event observable inside living cells. Rare events resist observation because each cell runs its own clock. Average, an asynchronous population, and every intermediate blurs into the next. Their move is to phase lock the population. A galactose switch fires the HL endonuclease at a single 18 base pair site in every cell at once, so one double-strand break, cut simultaneously everywhere, marches the whole culture through repair in lockstep. Each step now surfaces as a band that appears and then vanishes on schedule. The cut, the 5' to 3' resection to single strand, the strand invasion, the primed synthesis, the product. The timing is read by Southern blot, slot blot, PCR, and chip. This is the chapter's coinage, in vivo biochemistry mechanism, read off the living cell by synchrony rather than by purifying parts. This section explains how one experimental move turns DNA break repair into something you can measure directly. Repair is normally invisible because breaks fall at random sites, at random times, in only a few cells. No assay can average over that chaos. The whole method is one move. Put a single meganuclease called HO under a galactose switch so it cuts one known locus in every cell at once. That one substitution, from random and rare to located and synchronous, is what turns repair into in vivo biochemistry. Now a southern blot reads resection as a rising ladder. A slot blot weighs the single-stranded tail. PCR catches strand invasion 30 minutes before product, and CHIT times each protein's arrival. Synchrony is the enabling primitive. Every readout downstream is just a clock reading the same population. Break the synchrony, and the whole chapter's biochemistry dissolves back into noise. This section explains what makes a double-strand break repair assay in a test tube count as genuine non-homologous end joining. A double-strand break is repaired by gluing two ends together. Pull that reaction out of the cell and into a tube, and the whole chapter turns on a single criterion, whether the tube still means it. Joining itself is trivial. What certifies bona fide NHEJ is not that the ends fuse. It is that the fusion collapses when you remove the pathway's own machinery. Inhibit DNA PKCs with Wortmannin, deplete XRCC4 and ligase C4, and the joining drops by 100-fold. An assay captures a mechanism only when its readout inherits the mechanism's genetic dependencies. Extracts that join ends without needing them are catching something else. The in vitro reaction earns the name by breaking exactly where the living pathway breaks. This section explains how to prove that a repair pathway is genuine biochemistry, rather than a cellular artifact. The method is to take the pathway out of the cell and then demand that it fail in exactly the place where the living cell fails. Budman and Chu rebuild non-homologous end, joining in a human whole cell extract. They then certify its authenticity negatively. Real NHEJ dies under Wortmannin or under anti-XRCC4. So any joining that survives without DNA PKCase and without XRCC4 and ligase C4, the discriminating readout is structural. The extract yields linear multimers, never circular monomers, because the DNA never leaves its protein cage. The joining is therefore intermolecular, and it is quantified by PCR across two defined ends. The criterion of the real thing is the dependency signature, not the joining itself. This section explains how yeast recombination proteins are studied by taking a molecular machine apart and rebuilding it. You purify each yeast recombination factor, then read its role from what its addition changes in a reconstituted reaction. That is the whole move. RAD51 is the core recombinase, the eukaryotic ortholog of Rec-A. It polymerizes on single-stranded DNA into a presynaptic filament that searches a chromosome for its homolog and invades, forming a D-loop. Every ancillary factor is named by the exact deficit it rescues. RPA clears secondary structure yet overcoats the DNA. RAD52 delivers. RAD51 passed that blockade. RAD54 translocates on duplex to drive strand invasion. Genetics names which genes matter. Only purified proteins assayed in vitro reveal what each one does. The chapter's gift is the method itself, reconstitution as dissection. This section strips the recombinase down to its single core act and shows why every so-called ancillary factor becomes predictable once you do. RAD51 polymerizes on the resected single-stranded DNA tail into a right-handed presynaptic filament. About 6 monomers cover 18.6 bases, and the DNA is stretched to a rise of 5.6 bases, and the DNA is stretched to a rise of 5.4 angstroms. That filament searches for a homologous duplex and invades it. forming a D-loop. Naked Rad51 does this badly, and each helper patches one failure mode. RPA strips secondary structure from the single-stranded DNA, yet, left unopposed, it evicts Rad51. Rad52 mediates, binding all three partners to seed the filament onto RPA-coded DNA. Rad54 translocates the duplex to open it into RPA-coded DNA. Rad54 translocates the duplex to open it and to recycle spent Rad51. The chapter reads as a purification recipe, but the reusable primitive is the mediator logic. Derive the factor list from what breaks when the recombinase acts alone. This section explains how photobleaching microscopy measures the movement of DNA recombination and repair proteins inside living cells. Biochemistry proves what a repair protein can do in a test tube. It cannot say how the protein moves inside a living nucleus, where chromatin and competing processes set the real kinetics. The chapter's move is this: tag the protein with GFP, then perturb one side of an invisible equilibrium and time the recovery. Bleach the fluorescence in a small spot, a technique called FRAP, and unbleached molecules flood back at a rate set by their mobility. A fast, complete return means free diffusion. A slow or missing fraction is protein that is bound and immobilized. Now induce DNA damage and the free-to-bound ratio shifts. The recovery constant yields diffusion rates, the mobile fraction, and the residence time at damage sites. Mobility read as a proxy for binding, a relaxation measurement. This section explains how fluorescence recovery reveals a molecule's motion inside a living cell. Kill the fluorescence in one spot, then time how it returns. The recovery curve is the molecule's kinetics. The rate of return gives you diffusion, and the fraction that never comes back is bound, immobilized. This is the perturb and watch relaxation primitive. You do not read the equilibrium. You read how fast the system heals, a wound you cut into it. GFP supplies the reporter inside a living cell, retiring the old burden of purify, label, and re-inject. The chapter's payload lands here. A repair focus that looks like a solid complex under fixed cell staining is really being sampled, proteins arriving and leaving. Colocalization means only that proteins share a location. Complexation means they actually bind. The repair focus shows the first and not the second assembly happens on site. A snapshot cannot see this. Only the recovery can, because only motion survives the bleach. This section explains how synthetic holiday junctions turn an impossible assay into a screen. An enzyme that acts on a rare, transient intermediate cannot be assayed against the intermediate itself. The natural holiday junction flickers into existence during recombination and never accumulates. So the move is to fabricate the substrate. Anneal 4 partially complementary oligonucleotides and a four-way junction self-assembles, stable in a tube, presenting the same distorted crossover a resolvase reads. Heterologous arm termini. Pin it against branch migration so it cannot dissolve. A homologous core lets the enzyme sample cleavage sites. Resolvase activity now reports as 2 nicked duplexes on a gel. An enzyme discovery collapses to a screen. The primitive is this. When the true substrate is unobtainable, build a defined mimic whose behavior is a readout, and the enzyme that consumes it becomes visible. This section explains how a fleeting recombination intermediate becomes a stable tool for finding the enzyme that cuts it. Recombination welds two duplexes together at a four-way crossover called the holiday junction. Life everywhere keeps a nuclease that cuts this crossover back apart with mirror-symmetric nicks so that DNA ligase can seal the products with no further processing. Here is the problem. The real junction is a fleeting recombination intermediate. It is hard to catch and hard to hand to an enzyme for study. The chapter's move is to manufacture the substrate. You build synthetic forearm DNA that freezes the crossover into a stable, purifiable object, and then you let that decoy do the detection. Whatever cleaves it symmetrically is a resolvase. The primitive worth keeping is this. To assay a transient activity, build a static mimic of its substrate and read the enzyme off what it does to the mimic. This section explains why the in vitro non-homologous end joining system matters and the logic that makes it convincing. A double strand break severs both strands at once so no intact template survives to copy from. Non-homologous end joining, NHEJ, answers by joining the two ends directly, tolerating mismatch instead of demanding homology. That tolerance is the signature, frequent nucleolytic resection, near random junctional additions, opportunistic microhomology. This chapter's move is reconstitution. A pathway is proven sufficient when its function reappears from purified parts alone. Here those parts are Ku, the DNA, PK catalytic subunit, Artemis, and the XRCC4 and ligase 4 complex, and what they accomplish is the joining of incompatible ends. That is the sharp test, because earlier crude extract assays leaned on compatible restriction cut ends, the easy case. The generative primitive is this. To establish a pathway, rebuild it from clean components outside the cell. Sufficiency is the reappearance of function. Homologous recombination copies a sister chromatid. NHEJ negotiates a scar. This part explains reconstitution as the proof of sufficiency. Strip a repair pathway out of the crude cell extract. Rebuild it from a defined shortlist of purified proteins, and if the reaction still runs, that shortlist is the pathway. Non-homologous end. Joining is the hard case. It bridges two DNA ends that share no complementary overhang to guide them, so nothing but the proteins can hold the junction together. Ma and Lieber show that a handful of core factors suffice to seal incompatible ends in vitro. Those factors are Ku, DNA PKCS, Artemis, XRCC4 with ligase, 4 plus a fill-in polymerase. PCR reads out the diverse junctions. The tell of a defined system is its honesty about the gap. Efficiency runs from 0.1 to 1%, far below the cell. What is missing names? What is still unknown? This section explains why studying RAG and HMGB1 proteins means purifying them and rebuilding the reaction from scratch. Reconstitution is the epistemic move. To see how a nuclease measures DNA geometry, you strip the reaction down to purified parts and rebuild it. RAG1 and RAG2 together form the recombinase. This is the component that reads the recombination, signal sequence, and cleaves it. HMGB1 does no chemistry at all. Its function is to bend DNA, and that bending is what lets two distant signals come together, synapsing into a paired complex that obeys the 12-23 spacing rule. The thesis is that specificity is a property of the assembled complex rather than of the enzyme alone. Because of this, the assay has to be purify first, then reconstitute. The primitive carries beyond this one case. Any recognition event that is gated by geometry, whether by a bent substrate or by a spacing constraint, predicts an accessory architectural factor whose sole job is to make that geometry reachable, doing no chemistry itself. This section explains how antibody diversity is generated, and it begins with the surprise that the machinery is a transposase caught and domesticated. RAG1 and RAG2 read two recombination signal sequences, synapse them, and cut. They nick one strand, then let the freed 3' hydroxyl attack the opposite strand by direct transesterification, sealing the coding end into a hairpin. That is, transposon chemistry redirected from mobility toward immunity. The 12-23 rule, which says to pair only signals whose spacers differ in length, is the constraint that keeps the join productive rather than scrambling. GB1 contributes nothing catalytic. Its role is architectural, bending stiff DNA so that the two signals can meet. The chapter's move is to purify these players and watch the complex assemble and cleave in a tube, the entire origin of receptor diversity reduced to a reaction you can pipette. This section explains how one experiment proves what senses a broken chromosome. When a chromosome breaks, something must feel it before anything repairs it, and this entry is the physical proof of what that sensor is. The method is reconstitution as subtraction. You strip the cell down to purified protein, then add components back one at a time and watch which combination fires. ATM is the kinase that raises the alarm. The miR11, RAD50, and NBS1 complex, known as mRN, is the transducer that binds a broken DNA end and flips ATM from dormant to active. Isolating both to homogeneity is what licenses the causal claim. In the tube, nothing else can be doing the work. Necessity and sufficiency established by controlled removal and controlled return. The break is the signal and mRN is the switch. This section explains how a signaling pathway gets proven by rebuilding it from purified parts. A double-strand break never touches the kinase directly. The claim only becomes proof when you can pull the whole sensor apart and watch it fire from purified pieces. That is the move here. Reconstitution as causation. ATM sits as an inert dimer until damage splits it into active monomers. But the split is not driven by the break itself. The mRN complex plus a free DNA end are the sufficient stimulus. So, Lee and Paul purify ATM and mRN separately, recombine them over linear DNA, and show that phosphorylation appears only when both are present. This is the same requirement seen in cells, now rebuilt from parts. The generative pattern, a pathway, is understood exactly when its in-cell dependencies survive purification.